There we go. Okay, let's go ahead and pick up where we left off. Look about right. Where we left off, we're talking about thyroid, right? So basically, some of the things that the thyroid gland makes. There's three things that it makes. One is called thyroglobulin, and this is a globular protein. And thyroglobulin is actually part of the thyroid hormone production system. And we'll take a look at the sort of the nitty gritty or the details of that here in just a second. But thyroglobulin is a globular protein that's made by the follicle cells. Now remember, the follicle cells, we'll see this here in just a second, are the cells that are associated with that little ring. Where we saw the thyroid tissue has those little follicle rings. The cells, those cuboid cells that are in the ring, are the follicle cells. So these are the guys that are making thyroglobulin. And then what they do is they secrete it basically into the colloid region, which is that center region. So here's your cuboid wheel, right? Those follicular cells. So they're secreting it and they're dumping it into that inner portion, that colloid region. And that's basically where it stays. And the important thing about thyroglobulin is it basically provides a source of tyrosines. Now, the reason why tyrosines are important is because this is basically where the iodine attaches to make your thyroid hormones, T3 and T4. Okay. Of course, that brings us to our next ones, which is T4, which is thyroxine. So the official name for that is the big one, right? Tetraiodothyronine, right? So tetraiodo, that's 4 iodines attached to 4 tyrosines. Where are these tyrosines? They're in the thyroglobulin. So if you look at the thyroglobulin, most of the protein is sort of like a big carrying case. And the tyrosines that are at the heart of the thyroid hormone are the ones that will be attached to the iodines. Right? So once you actually attach your iodines to those tyrosines, then all you have to do is figure out how to separate that from the rest of the protein. And that's we'll see that here in just a second when we actually go through the mechanism of how these follicle cells make T4 and T3. Now, the other one is also T3, which is also an iodinated thyronine. It's a triiodothyronine in this particular case. So this is T3. It only has 3 iodines attached to 3 tyrosines. So the 2 of these are basically what makes up what's called your thyroid hormones. And these are the ones that are going to be released to the bloodstream to go communicate to target cells. Now, just kind of a little bit of an anatomy roundup. So just to kind of let you guys know. So here is your thyroid cartilage right there, right? That's the big gully whopper thing. And then of course you've got your cricoid cartilage here. And so these are basically this area right in here. So if you actually feel the upper ridge of your thyroid cartilage, you kind of go down, you can feel like a little bit of a depression right there. And then another big ring right there. That's this area right there that you're looking at. That's between the thyroid cartilage and the cricoid cartilage. If you're basically doing like if somebody's choking or something like that, and you have to basically set an airway into the trachea directly by puncture, that's where you're going to go right there. You're going to go in between 2 cartilages because it's stable and you got that little soft spot in there and it's easy to find. The little soft spots in between the cartilaginous rings are considerably smaller, so they're a little harder to get to. So you can actually feel that little dip right in there and you just go right in there. And then you'll be able to breathe. Of course, that's neither here nor there because we're looking at this guy right here, our big, beautiful butterfly. So you can see the body of it. That's going to be the isthmus of the thyroid gland itself. And then, of course, you have the 2 lateral lobes or the wings of the butterfly on either side. And that's going to be sitting just right about there. So just below that cricoid cartilage area. So right around in there. And you can see the other thing I want you to pull out of this one is the vasculature. So notice there's, no shortage of blood vessels that are going to this. which bespeaks its importance in terms of its need to basically distribute this hormone throughout the rest of the body. So you would expect, and typically this is true, most endocrine glands are very, very heavily vascular. Okay? So if you're cutting around in the thyroid, expect it to be a bloody business. So let's take a look then at the histology of it. And this is what your slides look like, which is one of the reasons why I like this Pearson product, because the slides are really close to what we have actually in the lab that we're working on. So first of all, here you can see the follicular structure, right? So for instance, you can see, I'm going to outline it right here, this little follicular ring. Now from 201, we can characterize that epithelial ring, right? It is simple cuboid, right? And then in the middle, you can see this is going to be the colloid region. And it's kind of like a semi-gelatinous sort of an area in the middle. That's going to be where you see your thyroglobulin being secreted. Now, these follicle cells that you see here, that is to say the cells of the ring, these are the guys who are making the thyroglobulin, the T3 and the T4, right? So that's what they're doing. But notice, I'm going to draw your attention though to another group of cells, the cells that aren't part of a ring. Right. So for instance, if you take a look at this kind of big mash of cells right in here, you can see it's in between two different rings. These are what's called the parafollicular cells, or the C cells. Now what these guys do is they're making something different. These guys are making calcitonin. And we'll take a look at that here in just a second. Of course, we all know calcitonin as being 1/2 of that homeostatic dyad that controls and regulates blood calcium levels in your system. And so this is where your calcium regulator is coming from in terms of calcitonin. And so in addition to this, you'll also see blood vessels. You can see here is the cartoon version. So I would use this to sort of decode the histology, but you can see here you've got your nice follicle cells. We'll take a look at that here in just a second in terms of what they're doing. The cells in between that are not part of the ring, these are the C cells or the parafollicular cells. You can use either one, right? So for instance, if I have an arrow pointed to, say, for instance, this cell right here, and I would say, what is that? You can say C cell or you can say parafollicular cell. They're synonymous. You do want to know both terms, however, because you'll see both used interchangeably. I tend to prefer parafollicular because it has more meaning to it. A C cell to me sounds like it's a C battery, right? And it doesn't mean anything to me. C, great. What did you do? That's just the third letter of the alphabet. You just randomly picked C. Well, not really. But parafollicular actually explains histologically and positionally, spatially, where you expect to find it, right? Parafollicular, next to para, the follicle cells. And that's exactly where you find them, right? So parafollicular cells will allow you to be able to sort of decode that. You can also see there's going to be some blood vessels in there as well, some capillary vessels, and then that's going to be all bundled up by the capsule, which is typically a connective tissue husk. And by the way, the capsule, you'll see this in a lot of organs, not just glands, you'll see it in kidneys. Most organs will have, if they don't have like a visceral lining system, will have some sort of a connective tissue husk, a capsule that surrounds them. In this case, you have that little capsule that's around the follicle cells or the thyroid. Okay, now let's take a look at the production of these hormones. So how exactly does a follicle cell go about the process of making T3 and T4? Well, first of all, what we're going to do is we're going to take a look at the activities of a follicle. So this is it here, this pink region, this is your colloid region. So that's the inside region of the ring. And this right here is your follicle cell. Okay, so we're going to be taking a look at what's going on in the follicle cell. And then next to it, adjacent to it, is going to be a capillary. Okay, first of all, let's start off from the beginning. The one thing we know about the thyroid is that it doesn't just make T3 and T4 whenever it wants, right? It only makes T3 and T4 under the direction of the pituitary hormone who's controlling all this, right? So the pituitary gland, the adeno hypophysis, if you will, right, or the anterior pituitary makes TSH, thyroid stimulating hormone, releases that to the bloodstream, and then that signals the thyroid follicle cells to make T3 and T4. So how does it actually? Basically, what you have in your capillary is, because it's bloodstream, you've got your TSH in there. So the way that the TSH actually regulates production is by using a TSH sensitive ion pump. Now, first of all, one thing to always keep clear in your head is that T3 and T4 are iodinated compounds. They are iodine tyrosine compounds. What that means is you need iodine, number one, which is the reason why you always buy the iodine supplemented salts. Right? And that's the only reason, by the way, that we have iodine supplementation in salt, because iodine is hard to come by otherwise in nature. Right? So, but here's the thing. How do I get a hold of the iodine? And how do I tell this cell to turn on, that is make T3 and T4 and release it, or to turn off, right? TSH has to have something to say about that. And it does. And the way it does is by basically turning on a TSH sensitive pump. So this is an iodine pump. This is like from general biology, basically just a pump who specializes in grabbing an iodine, pulling it from blood circulation in the capillary, and then moves it into the follicle cell. However, this pump is only activated in the presence of TSH. So if you don't have this pump activated, then you're never going to have the iodine in order to make T3 and T4. That functionally turns off the thyroid cell. So then in comes TSH. Why? Because the pituitary gland said, make TSH, we need more thyroid hormone. And then it's going to bind to this iodine pump and it's going to turn it on. Once it turns this iodine pump on, then the iodine that you've been consuming in your diet is going to basically be pumped into the follicle cell where it's going to diffuse. So the dotted lines are diffusion. It's going to diffuse throughout the cell. And then one of the things that's going to happen is now you have a reservoir of iodine inside your follicle cell. And that actually takes on a particularly interesting character, which is iodide. So it becomes an ion, essentially. And so when it moves into the follicle cell, what you have to do is you have to figure out now that you've got it inside the cell, you have to figure out how to actually take this iodine and bind it to the tyrosines. Where are the tyrosines? We learned about this on the previous slide, right? The tyrosines are bundled up in thyroglobulin. So we have to be able to merge it with those tyrosines and thyroglobulin, and that's exactly what happens. So we have basically a chemical process here, a mechanism, where the thyroglobulin will bind to those iodines at their tyrosines. So now what you have is tyrosines that are bound to iodines. And you have like a big bunch of protein here, and there's another tyrosine here bound to an iodine, and so forth. And that's going to be your iodinated thyroglobulin. So now at this particular point, once you actually do that, you have, I'm going to just put like a little subscript I there to just look So now what happens is you've got all the components of T3 bound to their iodine, and all the components of T4 bound to their iodine. So what it looks like is your T3 and T4 molecule are kind of like sort of encased, if you will, inside the thyroglobulin. Not unlike those like little excavation toys that they're making now for kids, where they give you like a little rock. It's like a chalk rock and like a little tool, and you kind of dig out the little gem, right? You're kind of excavating it. The gem is bound up inside the rock, and your job is to dig it out, right? It's just like this, right? T3 and T4 are like those little gems that are bound up in the thyroglobulin. Now, why would you do this? The reason why you would do this is because, number one, once it's actually bound up, you can secrete this into the colloid, and at this particular point, this can be stored for later use. This is really important, right? Because this is basically your storage strategy. It's always helpful to have a little bit of T3 and T4 on the ready, right? You never want to be caught flat-footed. And so if you have some thyroglobulin that's being stored in the colloid, then you're ready to start disseminating this as soon as the need arises. Well, what happens if that need arises? Let's go ahead and see how you release it, right? So this side right here, if you were to take a look at this side right here, you could see this as the storage mechanism. Making of thyroglobulin and storage. And on this side over here, this is the release. So let's take a look at this. We need more T3 and T4 in the bloodstream. So how do we do this? Well, the first thing we do is we take up some thyroglobulin by endocytosis back into the cell from the colloid. At this particular point, then, what we do is we fuse a lysosome. What does a lysosome do? Anybody remember what a lysosome does in cell biology? What? Yeah, they're digestive, right? So if you recall, what's inside a lysosome, which is in every cell, are hydrolytic enzymes. Hydro, water, lytic, to cut, water cutting enzymes. These are digestive enzymes. What they do for a living is they cut up all the macromolecules that compose you, right? So all of your nucleic acids, your proteins, your carbohydrates, and your lipids, they chop everything up into what? Into basic building blocks, right? It's like deconstructing a brick wall into individual bricks. So what you do is you basically have this endocytotic vesicle now with thyroglobulin. I'm going to just put a T in there for that. And then it fuses these two together. So just like two soap bubbles come together, when they touch, they fuse together. So they fuse together. Now what happens is you have your hydrolytic enzymes in your lysosome bound and sort of swimming around your thyroglobulin. So what is this going to do? This is going to digest the thyroglobulin. But not the T3 and the T4. Just like when you're carving out that little gem, you're sort of destroying the rock, but not the gem, right? The gem is saved from that. Now, as you're digesting this thyroglobulin, what you're doing is you're taking the protein and you're reducing it down to amino acids, and then you release those amino acids to make more thyroglobulin. But the problem is you've kind of lost a few tyrosines, haven't you? So you're always supplementing this process with more tyrosine so that you don't run out of tyrosine. Okay. So two nutritional metabolites that you need in order for this to work: iodine, number one, and tyrosine, number two. Okay. So if you get to be tyrosine deficient, then you can expect that to have an effect on your thyroid hormone production. So then once you digest all of your thyroglobulin, you've liberated your T3 and your T4. Now at this particular point, it just simply diffuses, moves out of the cell by diffusion and into the bloodstream, where it will get bound up by shuttle proteins whose job it is to grab a hold of T3 and T4 and move it around the blood cell. Remember, T3 and T4, tyrosine in particular, is hydrophobic. It's one of the hydrophobic amino acids. One of the non-polar ones. And so it needs a little bit of help getting around in this sort of water-based blood system. And so this is an example of one of those shuttle proteins. Remember we mentioned last time that a lot of what's in your blood plasma are these shuttle proteins designed to sort of move lipids all over the place. This is one of those examples of shuttle proteins that are designed, in this case, to move your T3 and your T4 throughout the bloodstream. Kind of makes for a fast and efficient delivery through the bloodstream itself. So this is fusion. There we go. So this is one of the things that we see now. One of the things I want you to focus on, because we see a lot of these. These are what I call the homeostatic cycle graphs. So usually two of them, right? One for the homeostatic peak, one for the homeostatic trough. But whenever we see these throughout the semester in just about every chapter. What this is designed to do is to sort of pull together this sort of regulatory cycle that we have on each side of every process. So let's take a look at this. Let's start off with our normal T3 and T4 concentrations. The first thing that we want to do is we want to knock it out of homeostasis. In this particular case, we're going to decrease the T3 and T4. The decrease in T3 and T4 is going to be sensed and communicated to the hypothalamus. And the hypothalamus is going to be like, oh, wait, we are dangerously low on T3 and T4. So that's going to stimulate the hypothalamus to make the thyroid releasing hormone, which we'll make, and it will basically release that to the hypothalamus. It will communicate to those pituitary cells to make and release TSH. What they will do, they will dump that into the bloodstream. That'll go throughout the body. The thyroid will pick that TSH up, and just like we just saw, that ion pump will be turned on by TSH. That'll allow you to be able to bind that iodine to the tyrosine, making essentially T3 and T4, which will then be released by the thyroid gland. And when it's released by the thyroid gland, it's going to increase your T3 and T4, bringing you back to homeostatic levels. Does that make sense? So on this side of the graph, what we noticed was there was a dip, a trough response. What we did was we basically made and stimulated more production of T3 and T4 in order to turn that back up. So this is basically a trough response. Of course, you can imagine that for every trough, there is a peak. And generally speaking, when we see these cycles, they come in pairs. So you see a trough response to turn it up. You see a peak response to turn it back down. Okay, so let's talk a little bit about these shuttle proteins because we did see them on the previous slide, right? There's 3 of them. The thyroid binding globulins are a big group of it. This accounts for maybe about 75% of the T4 and about 70% of T3 that's in the blood system. So a lot of the T3 and T4 is being carried by these TBGs, which is a large family. There's different subsets of these thyroid binding globulins, but basically what they do is they move T4 and T3 around in the bloodstream. Now, what about the other, the other quarter to other 30%-ish or so? What about those, right? So what about that T3 and T4? Where does that go? Because that's a pretty big percentage to just leave behind 25 to 30% of your T3 and T4. Well, the rest of it is mopped up by what's called transthyretin. This is basically a thyroid binding protein that is considered a pre-albumin. So albumin itself is a major shuttle protein. That shuttles many types of lipids. So doesn't albumin move around? Yes. Is it the only thing albumin moves around? No. Albumin basically kind of specializes on lipids in general, and it will move stuff around that are non-polar and hydrophobic in general. Now, when you take a look at a thyroid binding globulin, this one is a little bit more specialized. So this one will only grab on to T3 and T4, whereas albumin will be a little bit more kind of a broad selection. So it's not quite as specialized. And then the rest of it. Then what we have left behind after this is essentially a very small percentage of T3 and T4. So free T3 and T4 in your blood plasma is very, very tiny. It's about, it's less than 0.5%, basically, right? So you have 0.3% roughly of T3, 0.03 of T4. So by far, like well and above, all of it is largely bound up in one of these 2 types of transport proteins. Okay. So our thyroid stimulating hormone, so I want to take a look about this one. So the absence, it's easy to turn off the thyroid. Basically, all you do is just turn off TSH. So essentially, the presence of TSH. activates the thyroid. So what does that mean? So that means the default mode is off. So the thyroid, for the most part, is turned off. It takes TSH to turn it on. Okay. Another way that it does that is by binding to receptors that are specific for TSH, right? In this particular case, those receptors are associated with that ion pump, right? So you can tether that ion pump to a TSH receptor, whose activity will be to turn that pump on. So that's all well and good. Now we've got release of thyroid hormone, right? So we kind of have moved along the step. We started off with the hypothalamus releasing hormones to the pituitary, who then sent the pituitary hormone to the thyroid, and now we got the thyroid who's now sending the thyroid hormones to the target. So that's all well and good, but the question is, what does it do, right? So what exactly is the outcome and the effect of thyroid hormones? Well, first of all, there's a couple of things that we want to make sure we sort of clean up before we actually get into some of the activities and the functions of the thyroid hormones themselves. When you're talking about receptors and reception, so this is the reception of the hormone. It's important to understand that when your hormone is nonpolar, like T3 and T4, they can basically freely move through the membrane. So when you're taking a look at the receptors for T3 and T4, the receptors are typically going to be inside the cell. So you can imagine then there's two different ways that you can do this. If it's a polar hormone, then you have to have a receptor on the outside of the cell, right? To relay that message. That's that signal transduction thing. Remember we talked about the cyclic AMP before and all those different types of methods of internalizing the signal through these really elaborate pathways. That's generally speaking a polar hormone because you need some sort of an external receptor to bind to the signal message, and then it has to relay that message inside the cell. But when you're nonpolar, you're free to go straight through the membrane. You don't need an external receptor to relay the message for you. Your receptors are inside the cell. Now, where are they inside the cell? Well, first of all, number one, they're in the cytoplasm. They can be free in the cytoplasm, so you can bind to these receptors, and then that can activate some sort of an activity or function, which would create a sort of a downstream cascade of different types of activities. The other big one is where you need your message to be unpacked, if you will, or operationalized. In this particular case, you're going to see receptors on the surface of mitochondria. Why? Because remember, we just loosely said that the thyroid's job is to set basal metabolism. Where's your basal metabolism coming from? The mitochondria. So it makes a lot of sense to have your receptors on the surface of the mitochondria, right exactly where the heart of your activities are going to be occurring. And then, of course, the nucleus. Why the nucleus? This is for transcription of necessary genes. You may not have all the proteins that you need in order to pull all this off. So you need to communicate to the nucleus, hey guys, listen, we're going to regulate our metabolism. We're telling the mitochondria to sort of start turning on all the factory machinery and all that sort of stuff. But the nucleus, you need to start training the workforce. You need to start creating a labor market that we can fill the mitochondria with. That labor market, by the way, is the proteins, right? And so that training message is coming from the nucleus. That's in the genes. So this is basically a message to the nucleus to start transcribing and making proteins off of the genes that would necessarily be active in whatever metabolism you're doing. So this is basically important. So your thyroid is really, really an important in a global type of an organism. It's clearly, obviously, important for us. It's certainly essential for kids, right? Because your basal metabolic rate is very high when you're young. Why? Because you're growing skeletally, muscularly, and nervous wise. So those all need lots and lots of energy. And so there's a lot of basal metabolism, right? So what is some evidence? I mean, most of us have gone through this. We've actually seen the effect of our thyroid, even though we don't think about it. How? Well, some of us who may remember this or not, but I remember it clearly. When we're growing up, we go through the notorious teenage years, right? And I don't know about you guys, but man, when I was a teenager, I was like a plague of locusts. I mean, you pretty much can eat everything that's not tied down. And even if it is tied down, you would eat it. I mean, I would wipe out the entire refrigerator, and then I'd come back for another refrigerator full. And I would like always be hungry. But I didn't really gain weight. Right. And that lasted maybe into my deep 20s, and then eventually things like that changed. Right. I can't, by the way, eat an entire refrigerator anymore. I can, but I'll pay for it. I can eat considerably less than a refrigerator. I'm still paying for it. I'm still working. Summer's my like workout time. So I give myself the spring off and try not to add too much holiday doughiness for me to work off, right? But like when you're a teenager, it's like, you know, you've seen teenagers like this. It's like they're just eating all the time, but they don't gain any weight. Why? Because their basal metabolic rate has been shifted upwards. So their engines are running hot. They're burning hot fuel. Why? Because their bodies are very aggressively and actively growing. They need that material for their skeletal development, their muscular development, their neural development. Now, when you become middle-aged, that all changes, right? So we get that notorious metabolic shift, usually a downward shift in your middle age, right? Where we realize, wait a minute, I can't really eat the way I did when I was a teenager and not pay for it. Why? Because your metabolic rate has been sort of downshifted, and it continues to downshift, right? As you get older, I mean, like if you've seen like grandparents or parents like in their 70s or 80s, I mean, they can eat like one little meal a day and they're good. Why? Because their basal metabolic demands have been downshifted considerably. And so there's a natural sort of a developmental cyclic behavior to this sort of metabolic shifting, depending on where you are in your development. So what does it do exactly? Well, let's take a look at it, right? So we know it's metabolic, so this should be a pretty good metabolic discussion, and we're not going to spend too much. I just want to point some things out first of all. I don't necessarily want to start at the top, but I do want to start with number 3. First of all, in addition to building long-term muscle and skeletal and stuff like that, in us, what it does is it's basically very sensitive to sympathetic stimulation, right? So what does that mean? What's sympathetic stimulation? Give me an example of sympathetic stimulation. Yeah, right. Letting the lion in the room, or better yet, letting the exam in the room, right? That's sympathetic stimulation. It's a stress response, right? So if the lion is in the room, what does your body start moving toward doing? What does it want to do? Eat, drink, and be merry? No, everything about your body is all about run for your life, right? Think about that. Running for your life requires what? It requires contraction, muscular contraction, right? Which requires energy. Remember the whole ATP driven, the whole myosin kind of sliding filament model, that kind of ATP driven model. So you need ATP in order to run like the wind. And in order to make ATP, you're going to need what? Oxygen. So one of the things that the thyroid hormones do is it elevates the levels of oxygen consumption and energy consumption, which is going to increase your body temperature. Now, in order to get that oxygen, it's also going to increase your respiratory centers. So you're going to start breathing heavier. Why? To get more oxygen. To do what? To actually contract and run like the wind. But that's all well and good. You can breathe all you want, but you need something to go with that, don't you? You need a heart to pump that product, that oxygen, faster and more efficiently throughout your body. So you're also going to increase your heart rate and the force of contraction of your heart, including your skeletal muscles, in order to increase your blood pressure, which distributes your oxygen throughout your body, which allows you to run like the wind. Actually, no, it doesn't. It actually makes more energy, which then allows you to run like the wind. It also increases blood cell formation, which makes a lot of sense, right? Because who's carrying the oxygen around your blood in the first place? The red blood cells. So not only do you have red blood cell increase to allow you to carry more oxygen, to take it to the mitochondria to make more ATP so that you can actually do more muscle contraction and run like the wind, but it's increasing everything. So basically you're breathing more, you're distributing more, you're trying to increase your carrying capacity for oxygen delivery with your red blood cells. And then of course, you are also turning over minerals in your bone. Why that? Why would you want to do mineral turnover in your bone? In this kind of, everything seems to be all connected, right now. But this one seems like an outlier, doesn't it? What does your bone have to do with anything? You know where it fits? Because when you demineralize bone, remember there's two primary inorganic components of your bony matrix, right? Phosphate and calcium, right? So this is basically going to liberate calcium, and we already know that calcium is the major mechanism or the signal for muscle contraction. So you need more calcium in order to get more muscle contraction. It doesn't do you any good if you've got all the oxygen in the world, you've got all the ATP in the world, if you don't have any calcium to actually signal those sliding filament models to sort of kick off, you're not going anywhere. So demineralizing the bone makes sure that your calcium reservoirs are sufficient to basically handle the increased workload. So that's basically what it does. Now that's an acute version, right? So obviously sympathetic stimulation is sort of like an acute sort of situation. Over a lifetime, developmentally, this can be spread out over time in a longitudinal sense as more of kind of like an overall global maintenance kind of a strategy at your particular developmental level. Okay? So you always have both. Remember, this is the nice thing about physiology, because you're not just looking at like, okay, I need to be able to pick this thing up right now, right? That's kind of what we did it with muscles. You need a muscle load and muscle tension to be able to pick up an object. But in physiology, you have both acute response, that is, I need to do this right now, but you also have longitudinal response that I don't just need to have this done now, but I need to maintain this over a long period of time. And that's kind of what makes physiology kind of interesting. Okay, so let's talk about the untalked about at this particular point, right? So we basically have the C cells or the parafollicular cells that we looked at before. Now, these guys are important. Why? Because they are making calcitonin. Now, if you take a look at calcium homeostasis, you have a rise, increase in blood calcium, which will then turn down and it'll trough. It'll rise and it'll peak and then it'll trough and it'll rise and peak and then it'll trough and so forth. So where does calcitonin fit in this sort of homeostatic dyad for calcium blood regulation? Well, what this basically does is calcitonin is a peak response. So this is where calcitonin lives, up here. So in areas of high blood calcium, when you have high calcium, it's going to have a couple effects. Number one is it's got to get rid of that blood calcium, right? So you got to bring that back down. One of the ways that it does is it does a couple of things. So it's going to inhibit osteoclasts. What do osteoclasts do? They break down the bone, right? And they release the minerals, calcium included, into the bloodstream. So if you inhibit osteoclasts, then this is going to slow down your calcium release from the bone, which makes a lot of sense, right? You already have a lot of calcium. That's not the direction you need to go. So it shuts this off. The other thing is it actually stimulates kidney dumping. So if you stimulate the dumping of calcium through the kidneys, then that should bring your blood calcium levels back down. Okay. So what's the trough? Well, the trough is the parathyroid hormone. So this is PTH down here. So this basically happens when your blood calcium levels are low. They're troughing. So the idea here is you want to increase your calcium levels. And so the parathyroid hormone is coming off of the parathyroid gland, which are like little pine nuts. So if you have the kind of the wings of the butterfly here, there are like 4 little pine nuts that are actually on the surface of the thyroid gland itself. A lot of you guys saw this in your histology slides, where you saw the follicular structure and you saw these big purple collections of cells. Those were the parathyroid. Now they're secreting parathyroid hormones, and they only have one type of cell, chief cells, that are basically making this. And what they do is they basically secrete this and they antagonize the effects of calcitonin. That basically means they are the trough response to calcitonin's peak response. So they're basically moving it in the opposite direction. So here's kind of where you have your parathyroid glands located. That's it. There's nothing more than that. Now here, this looks familiar, yes? For some of you guys, because you saw this. I saw some of you guys see this. So here you can see you got your follicular rings here, right? So you got those there. You have this sort of mash of cells in between the follicles. Those are the parafollicular cells, the C cells. And this big guy right here, that's your parathyroid gland. And basically all those cells in there are chief cells, and they're basically secreting parathyroid hormone and dumping that into the bloodstream. So this is kind of like just a blow up of that. So in this particular case, there are 2 different types of cells here. There's what's called oxyphil cells and chief cells. I'll never have you try to differentiate that. Like, you know, get a close up and be like, okay, which one is the chief cell? Which one's the oxyphil cell? I wouldn't do that to you, because we need to have better slides and a better staining protocol in our slides in order to be able to really tease those apart. Okay, I'm thrilled if you can tell me it's a parathyroid gland when you see it. Okay. Okay, so that's good, but what does PTH do? Well, you would imagine it, first of all, it antagonizes calcitonin. So whatever it does, it basically increases blood calcium levels, right? So that's what it's going to do for a living. And so by doing that, the first thing that it does is it stimulates osteoclasts, right? So that basically releases calcium into the blood, right? Because the osteoclasts will break down the bony matrix and release the calcium into the blood. It also inhibits osteoblasts, which are basically doing the opposite. And what they're doing is they're taking calcium and putting it into the bone. So you shut that one off and you stimulate the mineral turnover, that is to say the osteoclast piece. So this accelerates your mineral turnover and the release of calcium. And it eliminates the deposition of calcium in bone, right? So this is the osteoblast shutdown. So that's number one. What else does it do? Well, the other thing that it does is it works with the kidneys to reduce loss. So it hangs on to calcium that's at the kidney level. So for instance, it communicates to the kidneys. It says, listen, you get a lot of calcium in your blood, don't let it go. If it's in the filtrate, right, that becomes urine, take it back, bring it back into the body, because we're a little short on calcium. We need to put that back into the bloodstream. So that's what it's doing with the kidneys. And then it also continues to work with the kidneys, not just for reducing dumping, so eliminate the dumping of calcium, but it also stimulates the secretion of calcitriol by the kidneys. And what calcitriol does is it basically enhances the absorption of calcium and phosphate in the digestive tract. So the calcium and the phosphate that you already have in your food, calcitriol under the direction of PTH will make sure that you are absorbing that with maximal efficiency. So a three-step system. Number one, the storehouse of calcium that you have in the bone, release it. That'll bring up your calcium levels. The kidneys, who will dump calcium in the urine stream, don't dump it. Hang on to it. That's number one. The calcium you have, hang on to it. And then number three is the calcium that you're bringing in, the new stuff, do a better job of getting it into the body. Okay, so it attacks your calcium blood levels on all three levels at the same time. So here is our homeostatic cycle, right? So we have our normal calcium levels under homeostasis. We're going to disturb homeostasis. In this case, we're going to increase our calcium levels. And so when we increase it, it's going to stimulate the production of calcitonin from the parafollicular cells, which will then stimulate the dumping for the kidneys of calcium. And it'll deposit that calcium into the bone, taking it out of circulation. So when that happens, that's going to increase your or decrease your blood calcium levels, which will bring it back down to homeostatic levels. Okay. Now, if you have a homeostatic deficiency, here you're going to disturb it, and you're going to have low calcium levels. Then what's going to happen is that is going to stimulate the parathyroid production, which is then going to cause the kidneys to hang on to calcium. That's what the reabsorption of calcium by the kidneys is. It will also release calcium from the bone by revving up the osteoclast, and then it will stimulate efficient digestion and absorption of calcium in the digestive system. All three of those will have the effect of increasing blood calcium levels, which will bring you back to homeostasis. And for you table people, here's your table. Okay, any questions on the thyroid before we move on to the adrenal glands? Okay. The adrenal glands. So basically, when you take a look at the adrenal glands, and this is a kind of a common sort of a thing, the adrenal glands themselves are, first of all, easy in humans to find. In cats, it's a little bit different, but in humans, it's super easy. I'll just draw here. Basically, you have your kidney that is shaped like a kidney, imagine that. And then, of course, you have your adrenal glands up here on the top. So that's basically where you have your adrenal glands. Oftentimes, you'll hear the term suprarenal. That's referring to the adrenal glands. So you'll hear that term as well. That's just a positional term. Supra, above, renal, the renal system, which is the kidneys. But the adrenal gland itself is actually composed of layers. The first layer, which is the outer layer, is the cortex. And by the way, whenever you see the term cortex and medulla, those are generic terms. And all that means is you're talking about an organ that has an outer layer, the cortex, and an inner layer, the medulla. And that's all that means, right? So the adrenal cortex is the outer layer, which itself actually has three different layers. And then you have the medulla, which is the core of the adrenal glands themselves. Now, the cortex is important. The different layers will make different things. So they make corticosteroids and various other sorts of things as well. And the adrenal medulla is largely focused on the adrenalins. So epinephrine and norepinephrine. And they're under the control of the sympathetic division. Remember, we saw that sympathetic nucleus in the hypothalamus, which had direct synapse to the medulla, so that you can relay your neural response of threat immediately to your adrenaline. What does adrenaline do? it basically upregulates your metabolism. So now we've seen a couple of things, right? Metabolically. So the thyroid is sort of setting that metabolic base. Then the adrenalines will basically upregulate you above that metabolic base in times of acute stress. So in reality, even though the thyroid is sensitive to sympathetic division, which you would expect it to be, it's not the one that makes you run for your life. The upregulation of your metabolism is largely coming from your adrenalines, right? Which is the reason why after the threat is over, you come down fairly quickly, right? Because that's the effect of the adrenaline. The thyroid would have a more persistent effect. Okay, so it'd be a little bit more persistent. So it'd be like, you'd be like torqued out for like a while. Okay. So let's take a look at the layers first of all. And what we're going to do is we're going to start from the outside going in. We're going to start with the cortex. So before we get to the three regions of the cortex, what we have above the cortex is the capsule, the adrenal capsule. So there's a little capsule where that's that little husk, like a little sack, like a little cellophane sack that kind of wraps around the adrenal glands. And then as you start to move inward, you'll start to move into the three different layers. The first one is the zona glomerulosa. So this is the outermost layer. The fasciculata, which is a large, very noticeable layer. I don't want to explain them too much because I want you to be able to see them, because it'll make more sense if we explain it while you're looking at it. And then of course the reticularis, which is a much smaller layer, and it morphs into the medulla. Now that's all great, but what do they all do? Well, they all have specific tasks, right? So for instance, if you're in glomerulosa, this is the outermost region, the first one. A lot of the cells here look kind of like grape clusters, and you'll see that here when we take a look at the histology. But what this one is actually producing is mineral corticoids. Mineral corticoids typically are steroid hormones that are associated with some sort of a metal homeostasis, right? Like an iron homeostasis or something like that, right? Some sort of a metal nutrient that's responsible for us. So these are metal-associated steroid molecules. The biggest one that we have is aldosterone. And what aldosterone does is it basically regulates sodium, which is a metal. And we're going to talk a lot about aldosterone because the activities of sodium are very critical in physiology. You've already kind of got the hint of that in AMP1, right? We're just looking at those sodium gates and how nervous impulses are transmitted with sodium. It gets even bigger. That story even continues. If that wasn't already big enough to win you over to sodium's side, there's still more for sodium. But this is a regulator for sodium. And so what it does basically is, this is one of those things, by the way, that we tend to talk about like throughout the semester, to be quite honest with you, aldosterone. And there's a really good reason for that. What it basically does is aldosterone basically allows you to conserve sodium. And this basically means that it increases sodium concentration in the blood. Now, that makes a lot of sense, right? Because if you're going to run a nervous impulse, you need those sodium ions to run your sodium channels, right? So this is going to be working to retain sodium in your system. That's what it does. It retains sodium in your system. It also kind of does a similar thing to what PTH does. It also increases your appetite for salt. So not only does it retain salt, but it also stimulates your taste for salt, which is one of the reasons why mammals are natural salt lickers. It also explains the reason why the snack industry is probably one of the biggest industries on the planet. Because if you want to get rich, invest in the 3 instinctual drives of human beings: salt, sugar, or lipids. If you invest in any one of those 3, you will make money hand over fist. Why? Because you have us over a barrel. We can't help it. I mean, we're going to go out there and we're going to lick on salt. I mean, look at the deer. I mean, they're licking salt, right? Look at us. We're eating a potato chip, which is little more than, I mean, are you eating that for the potatoes? It's like, you know what? Potatoes are healthy. They taste good. Does it matter if it's a potato? I've got a hypothesis. I think that if we took cardboard, deep fried it, and salted the devil out of it, we would still eat it. You know that's true. Probably somebody does that in the South somewhere. I mean, they do all sorts of crazy stuff, boiling their peanuts and fried pickles. I mean, they do stuff like what? I'm not a Southern guy. They sound good to me. I love pickles and I like fried stuff. So the 2 make sense. Boiled peanuts was confusing to me for a while, but I actually liked them. I tried them in South Carolina. I thought they were quite good. I like peanuts anyway. So there's really not a bad way you can present peanuts to me, but it's just weird, right? I mean, it's just kind of like, I would have never come up with that one. I mean, I would have been like, hey, why don't we just fry that pickle? Like, huh? It's like, fry a piece of cardboard, put salt on it, and somebody in the South will eat it. By the way, they're not the only animals on the planet. I'm just picking on the South because I like to pick on the South, but what? Yeah, right. So, yeah. So, but all of us will eat it. You sell cardboard chips and we will all eat it. Why? Because it's one of our major cravings. That's why McDonald's is the most evil, diabolical, hell-infused institution on the face of the planet. All fast food is, because they do the trifecta, right? Because what do? They give you those greasy lipids, salty, salt, fries that you can't not touch. And then what do you chase it down with? Yeah, a Coke. So they do the trifecta. Yeah, so that's evil. That's the reason why anybody who even gets close to McDonald's, even the worst one on the planet, is making money hand over fist and always will. Right. So if you're angry at McDonald's and you're looking forward to them closing, move on, because you are not going to win that battle. Right? We are just too much mammal and not enough cause. So that's just the way that breaks down. But this is it, right? This kind of gets to our, and it's the reason why I like to point this out and say that is because when you look at those 3 major cravings, some of which we really castigate. I mean, it's like, how dare you eat a donut? You're on a diet, blah, blah, blah. And then all of a sudden it starts to spin into hate and that sort of stuff. And it's like, usually it's just jealousy, because it's like, I don't have a problem eating a donut and you wish you didn't. Right. So that's usually what it turns into. But that, I mean, we really vilify people, right? We savage them if they eat donuts and stuff like that. It's like, you need to eat better. Well, yeah, I do, but I'm a mammal, right? I mean, that's kind of what's going to happen. But this kind of, I like to pause here a little bit to sort of expand on that a little bit, to sort of drive home the point that when we have our major cravings, these aren't an issue of willpower, right? I mean, whatever nutritionist or diet person ever came up with the concept of willpower, I want to actually punch them in the face, until they stop breathing. Whoever originally came up with that, I want to eliminate them from the planet. Why? Because there's nothing more psychologically, emotionally, and soulfully damaging to somebody than to basically tell them that they are a substandard piece of trash of a human being because they ate a donut. You know what? Newsflash for you. You're not going to beat instinct. Mammals are instinctually wired to eat lipids. Why? Because they are high in energy. They are instinctually wired to fatten up. Why? Because there's a famine right around the corner. You don't know when your next meal is coming. Guess what? Those of us who have a little portage on us are going to survive the famine. Those lean, mean, angry machines that basically castigate everybody, they're going to die. So you're not going to beat your instinct. So to basically denigrate another person like that for something that literally nobody can control, including the person doing the denigrating, is like beyond evil. Because we are wired physiologically to crave these things. That's just the way we're wired. We're wired to crave salt. Why? Because if you don't have sodium, you are dead in the water. So you are wired to crave salt. That's why it is one of our major cravings. You're wired to crave lipids because it's a survival strategy. And of course, sugar, same thing. It's also an energy thing, right? So it's a survival strategy. So I like to expand on that because one of the biggest issues that I have in all the healthcare industries, the biggest one I have an issue with is nutrition, quasi-nutrition, right? There's good nutrition, and then there's hacks and quacks. Unfortunately, the hacks and quacks are the ones that are the most visible and the loudest. The good ones are quiet and diligent and do a lot of good. I always tell students, a good nutritionist rarely has a loud voice. They have a quiet, confident voice and a wise one. When people write books, throw them out. Because I always say there's nutritionists and then there's people that write books. People that write books, as soon as they write a book, I eliminate them as a valid intellect. Because I've never read a book that wasn't basically infused with quackery. And that's unfortunate because nutrition is a really, really, really important topic. Unfortunately, we've got a lot of noodle heads in the middle of it. And they have no clue what they're doing. I mean, I've heard some ghastly things coming from these people. And I'm like, how are you human? I mean, honestly, how can you live with yourself? Oh yeah, that's right. Because you sold a bunch of books, you duped a bunch of people, you sold a bunch of books, and now you're basically floating on a yacht while other people are in counseling because you made them feel like a piece of crap. Bravo. Anyway, by the way, that gets a lot angrier, that soapbox. That gets a lot angrier and a lot bigger. I'll spare you that one, but only for a time. I have no problem going off on the healthcare industry. There's plenty of stuff to go off on. And it needs to be considered. Right? So that's one of them. So what does it do? So secretion of aldosterone basically is going to be tied to your sodium homeostasis levels, right? So for instance, if your blood sodium starts to drop, then you need to bring it back up. So that's where aldosterone kicks in. So it's going to basically start to increase your salts. Now, here's the thing. Why do you need to do that? Why is it so important? Well, this is important because when you increase your sodium blood levels, what you're also going to be doing is increasing your blood volume, which increases your blood pressure. Okay, wait a minute. Wait. So wait a minute. So you had me with sodium. I mean, that made sense, right? Because I did pass 2101 and I did understand the importance of sodium when it comes to action potential and stuff like that, right? So I was already there. I was already on team sodium, but now what is this black magic? I mean, what is this? I mean, how does sodium have anything to do with blood volume and blood pressure? Well, here's the deal. This is actually the reason why this system, aldosterone in particular, sodium specifically, is so critical, and the reason why we keep talking about it throughout the semester. This is only the first of many discussions we're going to have around aldosterone and the activities that are associated with aldosterone and its family. Well, here's the reason why this is so critical, because when you take a look at sodium, not only is sodium the major ion for action potential, but this is basically the major osmotic lure. For water balance in the body. What does that mean? Half of you guys are like, no clue. No clue what you mean by osmotic lure. Right? How about osmosis? This is one of those general biology ideas, right? We talk about osmosis. That is the diffusion of water, right? In osmosis, which is a key piece throughout all the body, there is one osmotic trigger phrase that helps you understand osmosis easily. 2 words. Salt sucks. Literally. So here's the thing. You're made mostly of what? Water, right? But here's the deal. Water, I always like to do like a little metaphor. I used to have a beagle. She just got old. But I always like to say water is a beagle. Now, I don't know about you, but you probably have a dog like this. Hound dogs are notorious for this. But beagles are notoriously stubborn. Very, very stubborn. Okay. So this beagle isn't going to do anything that I want her to do just because I want her to do it. Like say, for instance, if she's laying on the bed upstairs and I need her to go outside because, well, she's stubborn enough to stay there and just do her little diddles right there on the bed, because that would just be more comfortable. That way she doesn't have to move, just have to roll over to the dry side, right? And then, of course, we have an entire bed to change. Thank you, right? So what do? I go up there and I yell at the beagle, and that works? No, beagle just stays there, right? So I can get ugly about it too. I can really yell at the beagle, sound very menacing and scary. I scare half the family. It's like, the beagle's not afraid of you, but we are, right? So it's like, does the beagle do anything? No, doesn't do a thing. Now, I could actually physically pick her up. Now, beagles, as many dogs are, they are food hounds. So we're talking about a beagle that was a little on the chubby side. So it was no joke to pick this thing up. And she was one of the bigger beagles, not a little bigger, little beagles. But not only that, but she was kind of a little rotund. So to pick her up was no simple matter. Not only that, but when you picked her up, she was like just, all over the place. You pick her up and she wasn't one of those dogs that just kind of melts in your arms and gets all cute and cuddly. She was all like flailing all over the place. And and you start swinging that body weight all over the place. And you're like, well, you're going to hurt me and yourself or both of us. And I don't know how I'm going to get down the stairs with this flailing overweight beagle. One of us is going to end up in the hospital or the vets, but something bad's going to happen. Okay, so that's not a strategy, is it? I can't pick this beagle up and just haul her down there. So she's stubborn as all get out. She's not going to do what I want. So what I mean by that is water is a beagle. What does that mean? That means that I want water to go from here to here. But guess what? I can't make water do that. Water does what water wants to do. The beagle does what the beagle wants to do. And none of that, but guess what? In that argument that you're having with water, move here, move here. Guess who wins? Water. Water always wins. Always. You know how you know that? Ask the Grand Canyon. how that went. When the earth had an argument with water, who won? Water won, everybody else zero. Given enough time, water always has its way. So here's the problem. If I want water to go from here to here, I'm not going to make it do that. So how do I move the beagle? Simple. Sodium is an Alpo snap. If you don't know what Alpo snaps are, they're obviously Alpo, right? But they're just like little dog biscuits. They're about maybe that big or square. They're little training biscuits, right? They're kind of supposed to be for rewarding your dog when you're training them and that sort of thing. So what I found out was I can get that beagle to do anything I want. All I need to have is an Alpo snap in my hand. So if I want the beagle to get off the bed, what do? Do I yell at her? Do I pick her up? No. All I do is wave the Alpo snap in front of her nose, and then all of a sudden she's mine. I can walk down the stairs and she's following me happily. I can go outside, I can throw the biscuit out there, she'll eat it, and then she'll realize, oh, hey, here's some grass. I'll go ahead and do my business. So strong is that motivation for that Alpo snap that I could literally take that Alpo snap, throw it into the jaws of a great white shark, and she will happily go right in after it, not caring whatsoever that that's the last thing she's going to do. And the shark would probably have a pretty hefty meal. Probably wouldn't have to eat for a while, right? So that's how strong the motivation is. So in our scenario, sodium is an Alpo snap. So how do I get the water to move from here to here? Using an osmotic lure. Salt sucks. So wherever I want the water to go, all I do is put a pile of salt there. What's going to happen? It's going to suck the water toward it. So I effectively put a salt pile right here. The water's going to move by osmosis to the salt pile because it wants to dilute everything. If I want the water to move now over there, I move my salt pile over there and water chases right after it. So wherever the salt pile goes, water will chase after it. That's how you move water around in your body, is by using sodium as a lure. So then what does that mean? That means then that if you're retaining sodium in your blood, the water's going to chase back into your bloodstream after the sodium, and that's going to increase your blood volume. Of course, your blood volume is mostly made out of water, so that is also going to increase your blood pressure. One of the strategies for increasing blood pressure is sodium. Why? Because it's your osmotic lure. Of course, we also notice that potassium is inherently linked to sodium. So you also get a rise in blood potassium as well, concentration as well, to kind of balance those two out, right? Because sodium and potassium are like balancers to each other. They balance each other out. So that's basically what's happening. So aldosterone, we're going to have a lot more to say about it. It's going to feel like that we talk about blood pressure all semester long, and we do. Because blood pressure is not just some simple metric. It is simple, easy to measure, but it has absolute massive, titanic, and far-reaching consequences throughout physiology. It's a big deal, okay? And we talk about it a lot. Okay, that's glomerulosa, making aldosterone and other mineralocorticoids. What about fasciculata? That's the next one down as we're sort of moving in. So fasciculata is going to be basically making what's called glucocorticoids. So glucocorticoids are generally things that are associated with glucose maintenance. And use and things of that nature. So an example of this would be cortisol, hydrocortisone, and corticosterone. And basically this will convert it to cortisone, which will then be part of, and we'll see some of the activities of some of these here in just a second. And these will be associated with some of that sort of glucose as energetically metabolic types of behaviors and things of that nature. It's under negative feedback control. Cortisone will feed back on itself and sort of shut itself down. And this is the one that's actually in response to ACTH, right? So you can increase your cortisol and your cortisone levels from your pituitary. So your corticotropin releasing hormone, which will be coming off your hypothalamus, will be stimulating the adenohypophysis to make ACTH, which then goes to the fasciculata, and that increases your cortisone levels, and that will allow you to regulate your metabolism and things like that. Glucocorticoids in general will be focused on glucose synthesis, so you can actually make glucose, a process called gluconeogenesis, and it will also be involved in glycogen formation, right? Both very important processes in your overall kind of storage and management of excess glucose and carbohydrates, right? So it's a little bit different in terms of what it does in things like insulin and glucagon. It also has anti-inflammatory effects, so it'll actually activate white blood cells. We'll talk about the roles of white blood cells here in the next chapter, and there's a couple of other types of immune system roles as well. ACTH doesn't just stimulate the fasciculata, it also stimulates reticularis as well. And what reticularis does basically is it produces androgens, right? So an androgen basically is testosterone. Which is kind of important. Think about this for a second. Does everybody have a zone of reticularis in this room? Yeah. Does everybody secrete and make testosterone in this room? Yeah. Yeah, we all have two sources. Well, there's two sources of testosterone. Everybody has one. So everybody has adrenal testosterone, men and women. But men have a secondary source, that's the testes. So the primary rush of masculinizing effects of the testosterone is coming from the testes. That's the reason why boys and girls tend to have similar body shapes and sizes and sort of a lot more similarity in their body types when they're prepubescent. And then when they get into puberty, that testosterone just jiggers everything, right? And turns males into dudes, right? I don't know, right? But that's kind of what it does, right? And then estrogen, obviously being produced by the ovaries, will feminize the tissues, and that's what creates the changes in the body. It's those hormones, but everybody has testosterone, which is necessary because if it weren't for testosterone, I mean, think about what are the effects of testosterone, right? I mean, aggression, right? That's the reason why males, I'm talking about not just humans, but mammals, tend to be the more aggressive of the two. The sex drive, I mean, the randy teenager is a real thing. I mean, it's just like obsessive. That's because of the testosterone, right? But females also have testosterone, right? Because females also have a sex drive and they need one. Because if the females didn't have that sex drive driven by testosterone, then there would never be any interest in mating, right? And that would be a problematic sort of a situation, because then it would be like, well, wait a minute, if I'm not interested in males, then we're never going to propagate the species, right? And that's always a problem. So this is what basically where we find it. So you can see you got your kidneys and you got your adrenal glands sitting right there on top, and you can see lots of vascularization going into those adrenal glands as well, and kind of dumping right into the abdominal aorta and the inferior vena cava. And we'll talk more about that. So as we kind of move forward in lab and we get into blood vessels, those will start to make more sense. We'll follow those a little bit. And so this is kind of like the cutaway of it. So you can see you've got your cortex, you've got your capsule here, the cortex here, and the medulla in the middle. So histologically, when you take a look at the cortex, this is kind of going from the bottom up. So here you can see the capsule. So that's that little connective tissue husk that sort of surrounds the adrenal glands. And then you can see the zona glomerulosa. So you can see these guys right here. They're kind of a little bit more circular in nature. They kind of have a little bit more of a grape cluster type of a presentation to them. That's how you can see and tell glomerulosa. Then when you get into fasciculata, you can see it's much, much larger, and the opening, it's more white. So it's not that the cells are larger necessarily. It's that the cells themselves take up the stain that we stain them with a little bit differently. And so where glomerulosa tends to sort of hold on to the stain, the fasciculata does not. And so it has this wider appearance to it. And then eventually what happens is we run into reticularis, which is this guy right here. And you can see the beginning of it, and it goes right up until about right there, actually. So this is reticularis. So you can see the beginnings of it right here. So notice how where you have cells that largely aren't holding on to stain, now all of a sudden you get into a zone where the cells are now holding on to the stain. It's a pink stain, and it's starting to hold on to it. So that's the beginning of reticularis. So it kind of starts to move forward. So you can see the sort of transition zone. And then kind of the right down the middle of reticularis is going to be this little strip right up here. So that's like right down the heart of reticularis. But you can see the transition. You can maybe, if you want to, break this down and call this like a pre-reticularis area. So it's kind of like where fasciculata is starting to transform into reticularis, right in this area here. But you can see the cells are pinker, they're smaller, they're a smaller layer of it, but you can see they're holding that stain a little bit more. And then of course you have the medulla up here. So you can see very, very different cell texture up here in the medulla. Much more opening areas, lots of areas that are open, and like almost sinusoidal areas, which are kind of like these open channels and things like that. So the medulla has a very different type of a cell look to it, a cell structure to it than the cortex itself. By the way, if you're wondering, that's what we're going for in our adrenal glands histology, right? Is to basically be able to sit down with our slides and then look very closely at it and then be able to say like, oh, okay, there's the glomerulosa, there's the fasciculata, there's reticularis, and there's medulla. Okay, that's what we're going for there. Now, what about the medulla, right? We talked about the cortex, but now what about the medulla? So the medulla, basically you have two different types of secretory cells. One of them will actually produce epinephrine, which is adrenaline. And this is a good chunk of the adrenal secretions, right? So anywhere between 3/4 to, almost 80% of the secretions of the medulla is epinephrine, and then the rest of it is norepinephrine or the noradrenaline. Both of these together are basically collectively referred to as the adrenalins. They have similar functions. So some of their functions do overlap, but they're not the same. They do have some non-overlapping functions. So they kind of work together, but they're not the same. So let's take a look at what they look at. And oftentimes we kind of consider them together as a group. And so let's take a look and see what they do for a living. So basically, the first thing they do is they take the glycogen reserves, remember that's your stored glucose, and they start to liberate it, right? They stimulate the liberation of those glycogen reserves. Why? Because you need glucose to go through metabolism to produce ATP in order to get the energy to do muscle contraction, which is exactly what you do if you're secreting your adrenalins, right? Because the idea is you need to run like the wind from the lion, and you need ATP in order to do that, okay? So there's an upregulation of this. And then, of course, you're going to accelerate the actual ATP production. So that metabolic production that's happening inside the mitochondria is going to be upregulated. So that's the upregulation piece, the acceleration that I was talking about itself. Fat cells, the fat that's in them, remember the fat is highly energetic. So the fat that's in them is going to be basically broken down into fatty acids. This is called fatty acid oxidation. So you're going to take the stored fats that are in the fat cells, and you're going to start breaking them down. Why? Because you can feed those into your metabolism. I believe fatty acids will move directly into the Krebs cycle, because the Krebs cycle uses a 2-carbon acetate molecule, an acetyl-CoA, and fatty acids can be nipped off 2 carbons at a time and converted into acetyl-CoA and fed directly into Krebs cycle, which is about right in the middle of that whole metabolic pathway that produces ATP. So you can get a lot of extra production of ATP through this fatty acid usage. The other thing that happens is once you basically get this activation, then the glycogen molecules that are in the liver are going to start getting broken down. And what happens here is it's going to release glucose. This will be released into the bloodstream. And a lot of this will actually go to, I mean, we think of it as muscle contraction, right? So, oh, we need glucose in order for muscle contraction so we can run from the lion. But guess what? Who's faster? You or the lion? The lion by a lot, right? I think we have to go a long ways to find somebody that we can actually beat, right? Okay, so who's faster, you or a dog? The dog, right? If the dog really opens up the burners, we're gone. Now, we have to dig around a little bit, right? Who's faster, us or a turtle? Okay, we win that one, but guess what? Are we afraid of a turtle? They're vegetarians, for crying out loud. Okay, so anybody who's eating meat is faster than us, bigger than us sometimes, certainly have sharper teeth than we do. They have sharper claws than we do. Wow, we're batting 1,000 here, aren't we? It's like, this is terrible. Is there anything about us that is impressive as a mammal? Physically, no, right? I mean, our teeth are blunt. We can't bite anybody. We don't have any horns to gouge anybody with. We're not fast. We don't have any claws. We have nothing going for us, except for what? Right? Not our cheek, right? It's our brain. Why do we outlive everybody else? It's not our speed or our strength that helps us to beat the lion. It's our brain. Because we can think faster and assess faster than any mammal out there. Because mammals are largely driven by instinct, but we are driven by intellect. So before the lion even enters the room, we already have a plan, don't we? We already know how to beat the lion, because not only that, we've already understood the lion's weaknesses, and we figured out a plan to exploit those weaknesses to our advantage. Okay. That all takes a lot of power. And so that's the reason why this glucose that's being released ends up feeding our brain. Have you ever heard it said that heavy concentration burns as many calories as light exercise? It's true. By the way, that's not a replacement for exercise. So be like, you know what? I don't need to exercise. I think a lot. It's like, no, you don't. I can check all the games on your phone to prove the fact, but because whatever you were thinking about, those apps are usually designed to like undo all that good. And I love them for that. But anyway, right, this is it. It takes a lot of power. And so you're feeding that. Because think about it. This is your ace in your sleeve, right? You don't want your brain crashing on you in the middle of the threat. That's what you got to have going. So this really feeds your brain. And of course, it's going to feed your heart as well, right? So you're basically obviously beating your heart's beating faster. You're distributing oxygen better and nutrients better and faster. And so these adrenalines will actually go after what's called beta receptors. And there's different receptors that norepinephrine will go after than epinephrine. And they will stimulate and increase the contraction of your heart so that you can get better delivery of all of these. So here's your table for you table people. Enjoy. Okay. Let's do a couple of pickup glands and the pancreas. So not just a couple, like they're not important, but just cover a couple of other glands, and then the pancreas is going to be the next big one, and then we'll start digging into chapter 19. The pineal gland is the next one. This is an easy one. So this is where it's going to be found. It's going to be found in the third ventricle of your brain. So it's kind of the top portion. So if you were to get into the third ventricle, remember the third ventricle is that kind of the stalk of the T. So if you were to look at your brain directly like this, you would see the two laterals, and then the bottom one would be the third. So this is 1 and 2, the two laterals in your third, and then the pineal gland is going to be up in the upper portion of the third ventricle. It's going to be composed of cells called pinealocytes, and their job is to basically make melatonin. So what does melatonin do? Basically, melatonin is your sedative. This is like a natural sedative. So it'll basically start shutting down and inhibit your reproductive functions. It will it'll protect you against the damaging effects of free radicals that are generally produced during oxygen metabolism. And they're also wired into your circadian rhythms. The circadian rhythms are basically your sense of sleep-wake cycles and night-day cycles, right? So your circadian rhythm has multiple levels. So it's not just your sleep-wake cycles. It's also governs whether you're diurnal or nocturnal, whether you're active during the day or during the night. That's part of your circadian rhythms a lot, and it also expands outwardly into your seasonal rhythms as well. Like for instance, if you're an organism that tends to go into torpor, that basically means kind of a winter season dormancy, then that is part of the circadian rhythm component as well. So the circadian rhythms is kind of like a very fundamental sort of a life rhythm that we run across. And so what melatonin does then is this puts us to sleep. Have you ever noticed that when it starts to get dark, you get sleepy? That's because your pineal gland starts to make melatonin. It's actually light sensitive. So when you turn the lights out, it'll start to make melatonin, and melatonin will start to shut everything down. It starts to kind of power yourself down, and then that kind of gets you kind of the beginnings of falling asleep. Right? It doesn't knock you out completely. It kind of gets you start on that pathway, because then what happens is your arousal centers have to power down, right? And that's where you actually start to fall asleep and kind of fall into your unconscious state. And so, but it's kind of like the beginning of that whole falling asleep moment. So like when you're kind of like late at night and it's dark outside or whatever, and you're getting kind of drowsy, that's your melatonin probably kicking in. So it creates the drowse so that you can fall asleep. Okay, pancreas. So let's take, now the pancreas has, remember, both exocrine and endocrine functions. We're going to focus on the endocrine functions now. In the digestive system where it lives, we'll talk about the exocrine functions, okay? But basically the pancreas is situated right below the stomach and kind of right in the little crook. So where the duodenum comes off of the stomach, it kind of forms a little bit of a turn. It kind of fits right there into that little bend right there next to the duodenum of the small intestine. Now, when we take a look at the pancreas itself, the exocrine activities, largely what we're doing here is we're creating these alkaline buffers and the reason why we're making alkaline buffers is to basically neutralize stomach acid. And it's also enzyme rich, and this is basically digestive enzymes. So that's what the exocrine piece is. Now, the cells of the pancreas that we see, the general cells of the pancreas, what we see is what's called the pancreatic acids. So these are the cells that are basically making these sort of alkaline buffers and these digestive enzymes. And then they get secreted into the stomach itself. So that's the exocrine piece. The endocrine part, however, is basically located in these little islands or these little pockets of cells called the pancreatic islets or the islets of Langerhans. And there's a couple of different cells that you're going to find in there. The two biggies that we're going to focus on mostly are the alpha cells and the beta cells. Now, the alpha cells basically are responsible for making glucagon. And we'll take a look at the role of glucagon here in just a second. And the beta cells inside the pancreatic islets are making insulin. And a lot of us know about insulin, right? Because we either maybe or have somebody in our family who might be diabetic and they have to take insulin, right? So this is basically about blood glucose regulation. So that's basically what's happening with the alphas and betas. Now, there's a couple of others, right? So the delta cells, for instance, are similar to the inhibitory protein for growth hormone, so the GHIH. So this is basically regulating the expansion or the development of the islets, right? So basically, the reason why you don't expand these islets out infinitely is because of this GHIH. So if you make more delta cells, then you're going to keep your islets relatively small. If you don't make the delta cells, then you're going to allow your islets to expand and grow and have more cells in them. And so you can kind of create this size effect. And then, of course, there's F cells, which will make pancreatic polypeptide, which has a role in digestion. And so we'll take a look at this in the digestive side of things. So that's why I said most of our attention is going to be on the alpha cells and the beta cells. So it's kind of what it looks like. So you can see there's really nothing that looks like a Gorton's fish fillet. That's basically right there next to the duodenum. So you can see here's that little bend of the small intestine, the duodenum, and it's kind of snuggled right there in the little pocket. Kind of looks like a little fish tail. Where you see in the middle is going to be your pancreatic duct right there. And then the body of the pancreas is going to be in the middle. So most of these cells are going to be dumping enzymes and buffers into the pancreatic duct. That's the acinine cell. And then the islets of Langerhans, or the pancreatic islets, will be making their secretions, insulin and glucagon, and dumping those in the bloodstream. Because remember, they're endocrine. And if you're endocrine, you're dumping into the bloodstream. If you're exocrine, you're dumping it into a duct, right? So that's basically your pancreas. And this is what it looks like histologically. This is a zoomed up version of what you guys actually seen in your slides for the pancreas. So here you can see out here, these are going to be the acinine cells. So all these guys out here, those are the acinine cells. These are the ones that are secreting those digestive buffers and those enzymes, and are loading it into the pancreatic duct. Then the sort of region right in here where you see this very distinct shift in cell texture, these are the islet cells. And in there, you're going to see alpha cells and beta cells, and they're going to be making insulin and glucagon. And of course, you can see you have these capillaries. Why? Because you're going to be offloading your insulin glucagon into the bloodstream because you're endocrine in nature. Okay. And so you might want to take a look at those slides today, just kind of refresh your memory, or now that you've seen these and sort of broken them down, to take a look at that so you can sort of see it with new eyes, if you will. So let's take a look at blood glucose levels, right? We never miss an opportunity to take a look at physiology, so let's take a look at it. So when your blood glucose levels rise, so what happens is your beta cells are going to secrete insulin. And then what insulin is going to do is it's going to stimulate the uptake of glucose by the cells, by target cells. So the target cells are going to take the glucose out of the bloodstream, and that's going to reduce your blood glucose levels, right? That's what insulin does. However, when your blood glucose levels trough, then you're going to secrete glucagon, and this is going to basically secrete the release of glucose by the liver. So go to that whole glycogen breakdown thing, and then releasing that glucose in the liver, that's what glucagon is going to do. Okay. So if you're hyperglycemic, then you're going to get an insulin response. If you're hypoglycemic, then you're going to get a glucagon response. Okay. So here's our cycles. Let's take a look at it. So here's our homeostasis. Generally speaking, normal glucose levels between 70 and 110-ish or so. So we're going to go ahead and disturb glucose levels here. So our homeostatic glucose levels, we're going to increase it. And we have increasing, it's going to secrete or cause the secretion of insulin by the beta cells. And they're going to have a couple of things they're going to be doing. Number one is they're going to basically increase the rate of uptake of the cells. So that's number one. The second thing they're going to do is they're also going to stimulate those cells to utilize the glucose to make ATP. So they're not just saying, here, cells, take up more glucose, get it out of the blood, but it's also going to be like, you know what? Why don't you burn that glucose quickly so that you can take more, right? So keep burning that, so it upregulates that. So that's the second thing it does. Then insulin will also, if that doesn't work and you can't clear all of your blood glucose at that particular point, then what it also does is it will force and cause the conversion and stimulate the conversion of glucose to glycogen, right? So this is basically storing into glycogen. That gets the blood glucose out of the bloodstream as well and gets it into the glycogen form, which is typically hanging out in the liver. Okay, so it's out of there and you have it stored so you can use it for later. Like if you still have a lot of blood glucose in your system, then it's going to start using some of that glucose for things like protein synthesis and amino acid construction. So use it in other ways. How else can we use glucose? Not just for energy. We got a lot of it to lay around. Do we need any proteins? Do we need any amino acids? We'll build some of those and that'll get our blood glucose out of the way. Okay, so you're looking for ways to use it. And then last but not least, you still have a lot of glucose around, then it's going to start moving it into the construction of triglycerides, right? Because the glycerol backbone is a glucose derivative. So it's going to basically store it as fat. That'll take it out because you can hang on to that as long as you want. And that'll bring your blood glucose levels back down to normal homeostatic range. Well, that's all well and good, but what if we do it the other direction? What if we have hypoglycemia? In this case, we're going to disturb our homeostasis and we're going to drop our blood glucose levels. So in that case, the alpha cells will secrete glucagon, and what the glucagon is going to do is this is kind of the opposite of what insulin does. So what it's going to do, first of all, it's going to start to stimulate the breakdown of glycogen to glucose and it's going to release it into the bloodstream, right? So that's coming from the liver. That'll increase your blood glucose levels. So that's number one. Number two is it's going to go after fatty acids. So it's going to take some of those fats in the adipose tissue. And it's going to break it down into fatty acids. That's going to increase your blood glucose levels because you can convert fatty acids, those little 2 carbon chunks, into glucoses. And then it'll stimulate the release, the synthesis, right? That's gluconeogenesis and the release of glucose. It'll make new glucose and then release that. All three of these will increase your blood glucose levels, which will get you right back to homeostasis. In a glucose homeostasis pathway, at the top, we've got insulin. At the bottom, we've got glucagon. So we are close to the end, but we want to take a look at the function of insulin and of course of glucagon and see what they're actually doing. And so this is actually a perfect place to stop. And then what we'll do is we'll start with insulin next time, and then we'll finish out 18 and we'll get a good hearty start on 19 for the purpose of the exam, which I know that many of you are interested in. Typically, the exams will always be on